In the previous lecture, I introduced the concept of stem cells. Stem cells, if they are pluripotent, can generate all cells in the body. A source of stem cells has historically been aborted fetuses, what we call embryonic stem cells, or pluripotent cells from the inner cell mass of the blastocyst of an IVF embryo. Both of these carry with them huge ethical concerns. Because of this, scientists came up with the idea of what are called induced pluripotent stem cells. As we'll see, an induced pluripotent stem cell can take a somatic cell, which is a terminally differentiated cell, and reverse that process through the reintroduction of transcription factors, which are otherwise only found in an undifferentiated proliferative stem cell. This is the paper in which this process was first described. The lead author of this paper, Shinya Yamanaka, received the Nobel Prize several years ago for this work. What you can see by the title of the paper is that it is the induction of pluripotent, in other words, it can form all cells in the body, from mouse embryonic and adult fibroblast cultures. A fibroblast is a cell from your skin by defined factors. And by defined factors, what they're talking about is transcription factors and certain chemicals that we call morphogens, which derive the differentiation of cells across a particular lineage. And down the bottom, just to reinforce this, the Nobel Prize that was awarded in 2012 to Yamanaka and another fellow by the name of John B. Gordon, who actually began work on specialization of cells back in 1962. So what have we got then? What we see is, here is our pluripotent stem cell. The pluripotent stem cell, in the presence of morphogens and asymmetric cell division, undergoes epigenetic programming. In other words, it starts to rewrite its DNA. It changes the methylation pattern of that DNA. It changes the methylation and acetylation pattern of the histones which are associated with that DNA. That change in epigenetic program leads to a differentiated cell. This involves the change from an expression of a set of transcription factors that maintains a proliferative, undifferentiated phenotype to the expression of lineage-specific transcription factors that defines that particular cell. So in other words, a lung cell, a skin cell, a neuron, they are all differentiated cells. They all express different collections of transcription factors that define those cells as either a skin, lung, or neuron cell. So what a induced pluripotent cell is that it takes this highly differentiated cell with its lineage-specific transcription factors and its epigenetic programming, and it reintroduces those transcription factors that were otherwise only found in the proliferative undifferentiated stem cell. These transcription factors are induced, these transcription factors are introduced into the cell, and the cell once again becomes pluripotent. It's not as easy as it sounds, and it took a great deal of work in order to find the right mix of transcription factors and morphogens to achieve this. So let's remind us about what epigenetics is. In the undifferentiated state, we have the expression of certain genes. We see that there are specific methylation patterns on the histones, shown here. The DNA is open through acetylation of the histone, and we see gene transcription. During differentiation, many of these genes, which are turned on in the stem cell, are turned off. We start to see changes in the methylation of histones and the recruitment of methyl binding proteins, leading to condensation of the chromatin. We also see the turning on of different sets of genes, which are specific for a highly differentiated cell. The epigenetic barrier then is the epigenetic programming that must be reversed when trying to differentiate an adult somatic cell into an induced pluripotent stem cell. As we've already heard in this lecture series, when we've talked about X chromosome inactivation or imprinting, epigenetic modification of our genome can be a very, very stable phenomenon. And the transition from an undifferentiated proliferative stem cell to a differentiated cell and the accompanying changes in the epigenetic programming is incredibly stable. So the epigenetic barrier is not something simple to overcome and requires specific combination of factors in order to do that. So let's remind ourselves about the histone code. What we see here, we have heterochromatin, highly condensed chromatin, no transcription, and transcription, of course, is euchromatin, where we have the opening up of our DNA and transcription. In moving from no transcription to transcription, we start to see acetylation and specific patterns of histone methylation. In moving the other way, we see histone deacetylation. We see the recruitment of repressive proteins and the methylation of DNA. Histone acetyltransferases turn gene expression on. Histone deacetylases turn it off. There are specific marks on histones that are associated with heterochromatin and the more open euchromatin. Lysine 27, trimethylation on histone 3, is a repressor, whereas lysine number 4, trimethylation, turns genes on. Multiple sites of histone acetylation are all activating marks of transcription. The switch between these different patterns of methylation and histone acetylation and deacetylation is controlled by multi-protein complexes that contain specific histone acetylases or deacetylases, methylases and demethylases. And importantly, it also acts in concert with DNA methylation as well. So in other words, specific enzymes add and remove acetyl groups from histone subunits, histone deacetylases and histone acetyltransferases. This occurs with changes in specific patterns of histone methylation associated with gene activation and repression. So what about the transcription factors that turn these differentiated cells back into a pluripotent stem cell? Initially, there were 4 of them. It may have changed a bit since then, but for us, these are the 4. They are OCT4, which is essential in early embryonic development, and it maintains pluripotency. SOX2, it's involved in the regulation of embryonic development and determination of cell fate. It maintains self-renewal in these undifferentiated cells. KLF4, cell proliferation, survival. MYC, in addition to being one of the most frequently dysregulated oncogenes in human cancer, it also controls the expression of hundreds of target genes. And it's incredibly important for cell proliferation in the cell cycle. So between these 4 proteins, or these 4 transcription factors, we have controls on proliferation, differentiation, self-renewal, pluripotency. This is the code which is required to take a somatic cell back to a pluripotent stem cell. So for some more definitions. Firstly, an exogenous gene is a gene that has been introduced into a cell. The most common way of doing this is using a virus. An endogenous gene, on the other hand, is part of the normal genome of that cell. It's part and parcel of the genome of the individual. And in most cases, we have 2 copies of each gene. A somatic cell is reprogrammed by the introduction of exogenous copies of genes, cDNA. Remember, cDNA is the DNA copy of the coding RNA. So we introduced cDNA that encodes the 4 transcription factors. So we add them back to the genome. Because of course, in a differentiated cell, the copies of these genes are silenced. So we need to introduce active copies of these genes to initiate the dedifferentiation process. And that's shown here. So here we have our pluripotent stem cell. OCT4 is on and it's not methylated. In the presence of morphogens and asymmetric cell division, OCT4 is turned off, its promoter becomes highly methylated, and we start to see the differentiation process. In order to reverse this, we now take our cDNA, which we insert into a viral vector. The cDNA vector expresses our transcription factors, OCT4, SOX2, KLF4, and MYC, and in doing so, it reinitiates the expression of the endogenous genes, including each of these 4 transcription factors. So in other words, the reprogramming of a somatic cell into a pluripotent stem cell involves the expression of the transcription factors, OCT4, SOX2, KLF4, and MYC. This is exogenous expression and is done by plasmid vectors or viral expression. It triggers the reprogramming of the somatic cell. Reprogramming is defined as the reversal of epigenetic programming of genes such as OCT4. We're reversing the epigenetic marks. Once that has taken place, the cell is said to be reprogrammed. In other words, it now becomes a pluripotent stem cell once again. Once this has happened, the exogenous transcription factors that we added back to start the process are no longer required. Many genes that were turned on in the differentiated somatic cell, of course, are now turned off. And this again is overcoming what is called the epigenetic barrier. So here are our different cell types. We have blood cells, hepatocytes, skin fibroblasts, for example. And we reprogram these nucleated cells, such as the skin fibroblast, by introducing into that cell vectors, which are carriers of the cDNA expressing our transcription factors, whether it's a plasmid, a virus, or proteins and other small molecules that can carry the DNA back into the cells. By reintroducing these transcription factors into our nucleated somatic cells, we introduce our induced pluripotent stem cells. And these can be used later for therapy, understanding the molecular development of disease, and so on. This is a vector that is now used in generating induced pluripotent stem cells. What we see is that it has a sequence which drives the expression of these cDNAs. We see the cDNAs lined up here. In addition, we have a Lin28, which is also another protein required for the generation of these induced pluripotent stem cells. So we have a CAG promoter, which we see here. This EBNA and ORI-P sequence here ensures that this sequence is maintained stably in the cell and proliferates with the cell. The promoter drives the expression of the cDNA, and as a result, we have a self-contained unit which will drive the dedifferentiation of a somatic cell into a pluripotent stem cell.